What if you could create a digital version of a person with cancer, their digital twin, so you could test which treatment would work best for them before ever going to the clinic? Recreate complex biological systems in digital form to test drugs and refine combinations. This is the type of work that members of the public got to see and hear about at the recent public showcase in the Systems Biology Ireland Research Centre UCD, which leads a number of research programmes, including the Precision Oncology Ireland Consortium.
The treatments we have today started in a lab probably 10, 15, even 20 years ago, and the ones that will happen for the next generation are being worked on right now.
So we came together in this Precision Oncology Ireland Consortium, and we're trying to do exactly what it says on the tin. Develop precision approaches where we basically try to find the best treatment for each individual patient.
As we moved into the laboratories, Walter shared with visitors that they would be following the path of a sample through each stage of the testing process, each step building on the one before. The behind-the-scenes tour then began with the first stop in the tissue culture laboratory. Guests were shown what cancer cells look like under a microscope and how cancer cells are grown in the lab to then be used for testing a drug of interest.
When we get a patient's samples, we isolate the cells. We put them into two different dishes. In one of them, the cells are untreated, and in the second one, we treat the cells with a drug that we're interested in. And we look how the drug affects the cell viability and the cell growth.
We then followed the samples to the wet lab, where scientists looked at what is happening inside the cells in response to the drugs they are looking at, using techniques like Western blots to isolate a single protein of interest.
When I combine these two drugs at the lower concentrations, I can see huge amount of response, which is what we need, because these drugs can be quite toxic. So to reduce the toxicity, we kind of find the synergistic combinations of these drugs.
We then headed to see some of the cutting-edge equipment used to measure not one, but thousands of protein changes in the samples at a time. These highly specialized tools in mass spectrometry can generate huge amounts of data at a time about how the cancer cells are responding to the new drug treatments they are investigating.
In the previous talk that you've seen, you can look at 2 or 3 variables at one time. In this case here, we can look at up to 5,000 or 6,000 changes at any one time. So we can use these machines as fishing expeditions, and then we can use other processes like Western blots to validate what we see here.
At our final stop on the tour, we heard about how bioinformatic researchers apply mathematics and computer science to huge amounts of data coming from the biological techniques, working out how the cells talk to each other, predicting how they will respond to the new drugs or combinations.
We use this data to develop models called digital twins that mimics how those systems behave in response to drugs.
Finally, to close out the evening, people could chat with the early career researchers about their projects and their exciting results and findings. They also heard about another innovative project led by Systems Biology Ireland called Let's Chat Medicine, where teenagers work with researchers to learn about personalised medicine and create educational graphics and videos on this important topic. Guests, many of whom have raised funds for cancer research, were inspired and more optimistic for the future after their visit.